Welcome everybody to my talk entitled Systems Engineering of Microphysiometry. I like to talk about some engineering aspects of microphysiological systems and give some examples how we approach them. But first, some facts about Celasys. We are a southern Germany-based SME. We were founded already 15 years ago as a spin-off from Technical University of Munich. And we are equipped with laboratories for electronics, electrochemistry, a machine shop, cell culture laboratory. And quite a few years ago, we adapted some ISO standards, 9001 and 13485 for the engineering part, and also GLP for the biological part. We have a long-standing cooperation with the German Animal Welfare Academy, and we are a provider of instrumentation and services for microphysiometry. So what is microphysiometry? That goes back to a paper from Science from 1992. So what you can see here, that's a cellular model. And what they did back then, they placed a pH sensor close to that cellular model. And those they were able to measure the vitality of this cellular model in a label-free manner. And that allows toxicodynamic measurements. We extended that a little bit. We are not only looking at extracellular acidification, but we are also looking at cellular respiration and at cellular morphology and transepithelial impedance measurement. So to do that, for the extracellular acidification, we implemented a pH sensor. For the cellular respiration, we implemented a miniaturized dissolved oxygen sensor. And for the morphology and transepithelial impedance, we implemented impedance sensors. So that's our biochip. It's made on a glass substrate, so it's optically accessible, and it can be sterilized by ethanol. So that is the core of our talk. So systems engineering, that's a systems engineering approach on that systems that we have here. So it's always in the center. So we have a cellular model. We have hundreds of cellular models which are available, and they are always interfaced by a biochip for readout, which is connected to optics or electronics. And then very important, everything is placed in a climate control chamber with a more or less sophisticated fluidic system and with actuators. Usually that is controlled by computer. And what we indicated here, that you want to have not only one channel, but maybe you have more channels for your system. And that is how our approach looks like. So here we have 6 chambers with 6 biochips where we can put the cellular models on. On the back side, we have a pump. And here we have different fluidic modules where we can, before the start of the experiment, place the cell culture media in different flasks, the flask with cell culture media and the drug under investigation. And for example, media for the control, everything mounted in the incubator. And here we have the control unit right beside it. So to have a look at the historical development of the cellular models, here we have that great comic from Plants et al., how it developed from the end of the 19th century. So first we had cellular monolayers, then they became more complex. We went into 3D models. We went to tissue inserts, then we interconnected them. And on that development way, the fluidic system changed from a static fluidic system to a dynamic fluidic system and became more and more complex. And I like to show a few examples from us, how we develop systems according to that value chain. So that's an example where we used cells in suspension. I do not want to go too much into detail here. The main point here is that we can use cells with a cellular model which has cells which are available in suspension. So here we build a connector to our biochip where we just have a cellulose membrane. So say we could fix algae. So in that case, we used algae to make an example for water quality testing. And what you can see here is the photosynthetic activity of those cells, meaning the oxygen production of the algae when we put on a light source. And first we have a nice oxygen production, but as soon as we put metamithron to the cells, which is a herbicide, the photosynthetic activity goes down, and if we remove it again, it goes up again. What is also nice here, that since we are using label-free sensors, we can monitor the dynamic changes. We can add a drug or a compound, and we can remove it, and we directly see the effect of that. So the next step in complexity would be to monitor a 2D monolayer. From an engineering aspect, that's quite simple because those cells just adhere to our biochip. So we just seed it to the biochip and connect it to the fluidic system. So here we used a simple L929 fibroblast mouse fibroblast cell line, and we added 9 concentrations of a drug. And here again, here we see the extracellular acidification rate, meaning the pH change, which is caused by the cells. And when we add the concentrations, we can determine, for example, an MRD50 value. So when the metabolic activity of the cells is reduced by 50%, and that might be an important parameter for that SDS. Next step here is to go 3D. So we grew 3-dimensional spheroids from G2 cells in our lab. We grow those. We had to invest a little bit more engineering capacity to the encapsulation on the chip. So one spheroid would not be enough to make a high enough signal to monitor it with our sensors. So we made a plate here where we could integrate 9 spheroids, and then we have a nice and a high enough signal. And what we can show here, it's just a proof of principle. We see here the acidification rate of those spheroids. So here that's the raw data, and that's the rate. And if we add a toxic compound, the value goes down again in a label-free manner. So we were able to show that we can also use spheroids here. The next step then was to use tissue inserts. So we partnered up with Martech, and we used their epi-intestinal model, and again modified our chip. So now a commercial available 3-dimensional tissue insert model can go in here. And what we did here, we changed the fluidic system a little bit. So we have a basolateral fluidic and an apical fluidic, and we also could create an automated air-liquid interface. So we can exchange the media below the tissue insert. Then we can add media on the apical side, but we also can remove it and leave it to the air-liquid interface. And only if we add media on top again, we can, for example, make a tear measurement across the barrier of that function. And I have to say, that's all in the incubator, and everything is automated. So nobody has to open the incubator to exchange some fluidics or to insert some chopsticks. So the fluidic system is automated, and the measurement system is automated. So to sort it in to the development, so as I said, systems engineering, we have the development of the cellular models here, and that's the development, how we went along that to keep up with the developments in the cellular models field. Here are some more details about the fluidic system which we are using. So we are using a commercial available pump, a peristaltic pump, 8-channel pump, which is computer controllable. So very nicely can adjust the flow. So usually we use a periodic flow type, meaning we pump 5 minutes, we stop 5 minutes, we pump 5 minutes, we stop 5 minutes. But of course, you can program it as you like. And here we have the fluidic module, and we integrated computer-controlled valves here. So you can put 4 flasks in here, pre-fill it with the medium you want to investigate, and then you can transport it to your cellular model with a compound, with a positive control, as ever you like. And I really want to stress that here. So we made a quite simple investigation how the differences between a static fluidic system and a dynamic fluidic system. So most of us will just have their cells in a cell culture incubator and feed them twice a week, which basically is static. But we know that we are human beings, and if we would just eat twice a week, that would be problematic. So we compared it on the system, how the proliferation rate is compared in a static culture and in the dynamic culture, meaning when we exchange the media every 10 minutes. And the proliferation rate is changing drastically. So that's a basic thing which should be considered when we talk about systems engineering. Just the exchange rate of cell culture media will heavily affect the vitality of the cellular model. Coming to a next quite important step when we set up our assays. Here is our so-called Celasys 8 assay. So we set that to 24 hours, so it will nicely fit into a daily work schedule, so to say. But what is really important is that when one sets up an assay, we have to include controls and we have to include replicates. So what we do see here, so as I mentioned before, we have a 6-channel system, so we can make 4 replicates. And here we start with a reference medium for 6 hours, then we add treatment medium for 6 hours, then again reference medium for 4 hours, treatment medium for 4 hours, and then a positive control toxic medium for 4 hours. Everything is automated, and since we are working label-free, we see the effect of the treatment medium, and we see the effect of the recovery. And since I just talked about controls, so here we have a negative control where we do not add the test medium. So we just expect that we have positive signals here all the way until the end. So in the end, when we add our positive control, the signals from the cellular model has to diminish. And if that's not working, something that with our assay is not correct. What we also do, we add a blank. So we do not use cells on that blank just to make sure that the treatment media does not interact with our microsensors. And having that, we have quite a robust assay with all the controls which are needed. And yeah, we can rely on the outcome of those assays. That is an example. So I have 2 case studies here. A simple example where we just changed from FBS to chemically defined media, so FBS containing media to chemically defined media. The reference medium is standard DMA medium with 5% FBS. And the test medium is here a mixture of DME with F12, but without FBS. And what we can see here in the metabolism, in the acidification rate, if we change from reference medium to treatment medium, there is no effect. So meaning that that medium would really work nice over all that time. But when we have a look at the impedance sensor, we can see that without the FBS, the impedance goes really down, like here in 10 minutes, and stays on a lower level. And when we go back to the FBS, it goes up again. So that's quite aligned with the theory behind it, because all of those proteins in the FBS. But that also shows that it is important to have a look at the metabolism and at the morphology. Which brings me to the topic reproducibility. We are really working with defined media, and we highly recommend to work with chemically defined media, not to use fetal bovine serum. not only because there are ethical issues, but also because there is really a scientific problem in working with fecal bovine serum, because it's not reproducible. There is batch-to-batch variations, and when you really go out of the university lab into the industry and you start to do do your assays in different laboratories, and there is different FBS, there will be different results. But there is a community, a growing community, working on alternatives on developing chemically defined media. So here you find a website where you can have a look for chemically defined media. And we also have a poster about that. So you are welcome to visit our poster and to develop strategies how to move away from FBS. That's the second case study, just a quick one. We made our Celasis 8 assay, and we found out that for the CACO2 cell line, DME mixture with a 50 to 50% DME F12 mixture with ITS worked fine in our Celasis 8 assay, so in the 24 hours assay. What we also did, we moved to standard cultivation protocols, so which is state of the art, how to move away from FBS to make tests over weeks and months with the newly developed media. That also worked. We saw that the cells need a little bit longer to grow, but they still grew continuously, I think over a year or something like that. And what is also important, at least for the CACO2 cell line, if they stay, or if they still create microvilli, so if they differentiate, and they also did that after 40 days of cultivation in our chemically defined media. So that is just to show, okay, what was predicted by our Celasis 8 assay in 24 hours seems to be valid. We controlled it in more than a half year of other experiments. So to summarize, the systems engineering approach I recommend. I gave an overview how those microphysiological systems can be categorized, and stated the importance of flow of the cellular model and the temperature, how important it is to have a standardized protocol. And with the automation, we also improve reproducibility because we exclude human errors or errors due to drops in temperature and something like that. I highly recommend to use defined media and to validate the cellular model, meaning to validate cell lines which are used. So that was a problem we had like 20 years ago with the cross-contamination, but we see that especially at universities, the validation of the cell lines is not common practice, so to say. And I want to emphasize that system engineering may contribute to the reproducibility crisis in biomedical sciences. But just wrote that paper, Systems Engineering of Microphysiometry, and I invite you to have a look there because there are some aspects are explained in more detail. And with that, I come to my last slide, and I'm happy to take any questions.
Thank you very much. Are there any questions from the audience? I have maybe more specific questions on the spheroid model that you have shown, that you have the measurement in the raw data. You had these waves of data. Can you maybe explain from the data recording from where they come and how the measurement actually looks like?
Yes, excellent question. Thanks for that. Usually I do not explain that because it's a little bit confusing and complicated and makes my talks too long, but it's really a central question. So what we do, we run our fluidic system in a stop and go mode, meaning we pump fresh media towards a cellular model for 5 minutes, and then we stop. And then in the next 5 minutes, we can see the acidification rate of said microvolume we pump there. And then we pump again for 5 minutes. So we have fresh media on the chip. We can use that to recalibrate the sensors. Then we stop again, measure for the next 5 minutes, and so on. And we program it that we have these 10-minute cycles for the first 6 hours, for example. And then in one of the pump cycles, we switch one valve. So we can add then the cell culture media with the drug under investigation. And that's, so we call it shark fins. So the highs is the acidification rate or the oxygen consumption rate. Yeah.
Okay, thanks. You mentioned that your system has like 6 replicates in parallel that you are using. Are you also planning to extend this to being able to have more replicates in the same system?
Of course, if you have a look at the graphics, where the graphic is going and what we developed so far. So of course, the next thing is a 24 well plate.
What are the challenges there for the scale up? What are the technical challenges there for the scale up?
I think it's, with our experience, it's just a logical next step. It's not that there are too high hurdles, I would say. Of course, you would use the standard format of a 24 well plate. You would use a pipetting robot. You place everything in a huge incubator. So we have a prototype on that. We are working on that.
And on the fluidic side, I mean, at the moment you use the peristaltic pump. Would it be then a different pumping system if you have much more wells?
Yeah, a pipetting robot.
Okay. Oh, okay. Yeah.